Hello grade 12 students. Today is Wednesday, March 18. We will start with a new section discussing mutations. Please make sure to open your books on page 372. The objectives of this section is to identify mutations and determine how they affect genes. We already know that the sequence of bases in DNA are letters of a coded message. For example, AGC are the letters that will later make an amino acid. But what would happen if a few of those letters changed accidentally? Could the cell still understand its meaning? To answer those questions, let's discuss mutations. What is mutations? When the cell makes mistakes in copying its own DNA by inserting the wrong base or even skipping a base is called mutation. Mutations are heritable changes in genetic information. That is, they can be passed from parents to offspring. So, as we have said earlier, that mutation is when the DNA changes its structure or sequence. There are two types of mutations that will be discussed. Gene mutation, which produces a change in a single gene, and then chromosome mutations, which produces changes in a whole chromosome. Let's discuss gene mutations first. Gene mutations, or we call them point mutations, involve changes in one or a few nucleotides. They occur at a single point in the DNA sequence. If a single gene is altered, the alternation can be passed on to every cell that develops from the original one. Point mutations occur during DNA replication. So let's say, if you have a sequence of DNA which includes nitrogenous bases, a gene will be mutated when one of these bases are either substituted, inserted, or deleted. Substitution is when one base is changed to a different base. Substitution can have no effect on a gene and will not change the amino acid. Example, if mutation changed one codon of messenger RNA from CCC to CCA, that is, adenine is replaced by cytosine, the codon will still give the amino acid proline. However, a change in the first base of codon CCC to ACC will replace proline with threonine. This figure will explain more the point mutation substitution. Please look at the fifth base of the original sequence. It is thymine. In the point mutation, it is substituted or replaced by cytosine. This substitution will still give the amino acid lysine and will not change the sequence. Insertion and deletion. They are point mutations in which one base is inserted or removed from the DNA sequence. The effect of these changes can be dramatic. If a codon is inserted or deleted, the bases are still read in groups of 3, but now those groupings shift in every codon that follows the mutations. This is called frame shift mutations because they shift the reading frame. Frame shift mutation can lead to proteins that are unable to perform their normal function. We have already discussed substitution. Now please look at the second sequence to the right. Take your time and inspect. See how cytosine is inserted after the fourth base and how the frame shift is moving forward. This sequence started with 11 bases, and after insertion, they became 12. To the left, you will see how thymine is deleted from the sequence, and how the frame shift is moving backwards. It started with 11 bases and ended with 10. This is why changes are dramatic in deletion and insertion, but not in substitution. Let's move now to chromosomal mutations. They are changes in the number or structure of a chromosome, and they are of 4 types: deletion, duplication, inversion, and translocation. So let's start with chromosomal deletion. It involves the loss of all or part of the chromosome. You can see here you have an original chromosome, and after deletion, how does it look like? Chromosomal duplication is when an extra copy of all or part of the chromosome is duplicated or doubled. While gene inversion is when part of the chromosome is reversed. Finally is chromosomal translocation. It occurs when part of one chromosome breaks off and attaches to another chromosome. What are the effects of mutation? Some mutations may or may not affect an organism. Other mutations can affect species or an entire ecosystem. Some mutations can be due to stressful environmental conditions or simply by an error in the DNA replication. Others arise due to mutagens. Mutagens are chemical or physical agents in the environment. Chemical mutagens include certain pesticides, natural plant alkaloids such as morphine or the plants that give ephedrine, tobacco smoke, and environmental pollutants. Physical mutagens include electromagnetic radiations such as X-rays and ultraviolet light. Cells can sometimes repair the damage, but when they can't, the DNA-based sequence changes permanently. The effect of mutations vary widely. Some have little or no effect, and some produce beneficial variations, while others negatively disrupt gene function. Harmful mutations can change a protein structure or gene activity. Some cancers are, for example, an effect of mutations that causes uncontrolled cell growth. Sickle cell disease, or sickle cell anemia, is a disorder associated with changes in the shape of red blood cells, whereby the oxygen carrier protein hemoglobin cannot function anymore. Not all mutations are harmful. Beneficial mutations can occur in humans. For example, mutations that increase bone strength and density, and others that increase the resistance to HIV. Polyploidy plants that have an extra set of chromosomes are considered larger and stronger than other diploid plants. An example of a beneficial mutation is lime. It is a seedless citrus plant that due to mutations, the size and the strength of the tree is affected, as well as the quality and the seediness of the tree. In this figure, you will see multiple polyploid plants that have extra chromosomes in their genetic information. End of section 13.3. Thank you for your attention.